All right, thank you all for joining us and welcome back to the next webinar in our Plantae Presents Global Plant Science Talk Series. My name is Katie Rogers and I'm your host for today's webinar. Before we get started, I'd like to go over a few things just to make sure you get the most out of attending today's webinar. This webinar series is brought to you today by Plantae, the open online community for plant scientists powered by the American Society of Plant Biologists. I'd like to give a special thank you to all of our ASPB members who are attending today. Your ASPB membership dues help support and make these webinars possible. For any of you who have not yet joined ASPB, you can join today and use the discount code PRESENTS10 to receive a 10% discount on your registration. ASPB members get early access to these webinars. You can learn more about our organization and the opportunities that we provide at aspb.org. Today's talk is the 15th Plantae Presents webinar in this series. If you haven't already done so, check out our archive to watch all of the previous talks in this series. You can find information about all of our speakers on the blog and subscribe to our YouTube channel. This season, we will be experimenting with a couple new formats. On the dates listed here, we will have shorter 7-minute talks featuring the work of 4 researchers. After their presentations, we will open a panel discussion. We hope that this new format will allow us to include even more amazing presenters in this series and make these webinars even more interactive. Visit our website to learn more and apply to be a panelist. If you're having trouble connecting or need to leave the webinar early, know that a recording will be made available along with all of the associated materials within a few days. Today's talks will each be 20 minutes, followed by 10 minutes for questions. If you have questions for our panelists, please let us know using the Zoom question and answer section. You'll notice that there's also a chat area open. This section is here for you to introduce yourself, say where you're from, and connect with your colleagues. Questions specifically for our panelists should be shared in the Q&A section to make them easier for Yasin Degas, our moderator, to navigate. All right, so I'd like to introduce our moderator today. Yasin is a group leader in the Gregor Mendel Institute in Vienna. He's here with us today to take your questions and read them aloud to our speakers. With this, I'm going to hand it over to Yasin to introduce our speakers and moderate the session. Thank you.
Thanks, Katie, and welcome everyone for joining us today. Today we have 2 amazing talks lined up for you that will cover the evolutionary aspects of molecular plant-microbe interactions. And the first speaker is Sophien Kamoun from the Sainsbury Laboratory, and he will tell us about NLR networks and immunity. Sophien, please.
Yes, thank you. Thank you, Yasin, for the introduction. I hope my slides are visible to all.
Yep.
Good. Great. So I'm really excited to be speaking today and to be part of this session. I think I have a couple of introductory comments. The first one, it's nice to be within my academic family because I feel that Yasin and Phil are kind of academic kids and grandkids. So so it's kind of fun to be share this session with you. And secondly, I think the topic today, the overarching topic is quite timely because I think our field of molecular plant microbe interaction has now embraced the evolutionary approaches, evolutionary methods. And so I think today's speaker, I know Yasin also shared these views and many others, are now realize the potential of bringing in an evolutionary perspective to mechanistic research. And this EVO MPMI approach is exciting to us because it can provide insights that you would not obtain if you're studying just a single gene or single protein. You really need the comparative approach to gain these insights. So my first slide is really a very general introduction that I hope many of you know about, but I think by now we know that pretty much all pathogens, all types of pathogens and pests secrete effectors to modulate host processes. What's relevant for today is that some of these effectors navigate inside the plant cell, and so they function inside the plant cell to help the microbe in various ways. They suppress immunity, but they can do also other things. But plants don't sit idle, and they have evolved receptors to detect these effectors. And in particular, I want to highlight the NLR type receptors, intracellular immune receptors that are of this widespread family that we find not just in plants, but also in mammalian systems, fungal systems, and quite others. And these intracellular receptors can detect the effectors. And in a way, when an effector is detected, it's now tripping the wire and activating immunity instead of helping the pathogens, actually blocking it. So this creates a very unstable evolutionary situation. And this probably is what resulted in this very large number of immunoreceptors in the plant, and also a very large number of effectors on the pathogen side. This tweet is meant to be provocative. I don't mean to say that these are the most diverse protein families, although they might be, but they're arguably among the most diverse protein families in the pathogen and in the plant, respectively. So I'll talk about NLRs, as Yassine mentioned, and NLRs were first cloned as essentially as the products of disease resistance genes, are genes, usually single dominant genes that segregate in plant populations. And they were cloned back in the early 90s. And that was a landmark event in plant pathology. That was really the sort of validating the Flores gene for gene model and really bringing a molecular genetic perspective to plant pathology and the plant side of plant pathology, really, in a way, perhaps even the birth of the plant immunity field. But what really turned out to be nagging is understanding how these NLRs function. So only in the last year or so, we started having really good insight of how they function. And this is through this so-called resistosome model. So the idea here is simply that these NLRs, which are multi-domain proteins with an MBR domain, central domain, a variable N-terminal domain in plants, these, whether they belong to the CC family or the TIR family, when they're activated, they oligomerize into this resistosome structures. And this process is essential for the activation of the hypersensitive cell death, which is the readout of these immune receptors. The main immune readout of these immune receptors, but possibly also other subsequent responses. And this is through work on first with the ZAR1 CCNLR. This is an Arabidopsis NLR, and this is beautiful cryo-EM structure from Gigi Chai and Jiamin Zhu in Beijing. And just more recently, a few weeks ago, Raoul Martin, working with Eva Nogales and Brian Staskowitz at UC Berkeley, posted the ROC1 structure, and this is a TNLR, and this is a tetramer, not a pentamer. ZAR1 is relevant to our talk today because the way ZAR1 gets activated is through essentially flipping out this N-terminal alpha helix, which is normally in the inactive state, buried within the CC domain, which is a multi-helical domain. So when it gets activated, when ZAR1 gets activated, the alpha helix flips out and it forms this structure that you can see on the right side, which end up being essentially now a pentamer of alpha helices. And this tunnel-like structure is presumably, it's not fully understood yet, but presumably inserts into the plasma membrane and causes a cell death directly. So it's not really a signaling pathway in the classical example of signaling pathways, but perhaps it's a direct perturbation through membrane perforation, a toxin-like activity by the ZAR1 resistosome that causes the hypersensitive cell death. As an aside, I'm going to be talking today about a couple of lockdown projects, let's call them, which are really computational projects we started when we had to shut down the lab back in March due to the coronavirus pandemic. This is one of them that was led by Yorgos Kourelis, and Yorgos essentially crawled the literature for plant NLRs that are experimentally validated. He has a database of 450. You can read his preprint. We're going to be posting the second version soon, I hope. What's really interesting here is first that there's really quite a number of plants where now there are genes and NLRs that have been cloned and validated, but you still see huge gaps here in terms of plant phylogeny. For example, there are no early diverging plants, so this is to relate this talk to Phil's talk, and there is actually huge swath of plant diversity where there's still no NLRs validated. So those of you who are inclined to do some sort of maybe evolutionary study or explore plant diversity, I really encourage you to look at some of the studied species and use this data set as a guide. Okay, so now I want to mention the end, really. I want to tell you about the take-home message. I think it's a lot easier that way. And I think the take-home message really here is about reconstructing the evolution of NLR proteins. And the model we came up with is that NLRs ancestrally were essentially, if you want, multifunctional receptors. So they have the capacity to detect effectors directly or indirectly, sometimes through host target, known as, or host partner, should I say, known as a guardee or a decoy. But these multifunctional ancestors can also execute the immune response on their own. They don't need additional NLRs to do that. And throughout evolution, these multifunctional receptors have specialized into sensors and helpers. And so the sensors are now dedicated to sensing the pathogen, sometimes through integrated domains or decoy domains that are essentially have been recruited and integrated within the canonical architecture to trap effectors, other times directly or even indirectly, but they rely on helpers which are now specialized into triggering the immune response and not being involved, at least directly, in detecting the pathogen effect. So this is the one model, but this is not the whole story. There are certainly examples of NLRs that are quite conserved across evolution and that have remained as multifunctional receptors throughout time, as shown here. And in this case, the diversification is happening at the level of the guardee or the decoy, not at the level of the NLR protein itself. So there are different NLRs have experienced different evolutionary trajectories, if you like, throughout time. And that's kind of the message really of this talk. So the question we were really interested in when we realized that there are NLR networks, we knew already there are NLR pairs for many years, and work in my lab resulted in finding some of the first extensive networks. We realized that we should really try to understand the molecular consequences of transitioning from a singleton to pair to a network. And this really brings these two fields together. So this is the network we identified, the NRC network. As you can see, the first layer is a number of famous R genes like PRF, PTO, RX, BS2, and so on. These were cloned in starting from the 90s. And for a long time, people really struggled in finding downstream elements. Well, it turned out that the downstream elements are these NRCs. So these are helper NLRs. And as you can see here, they function redundantly to work with some of these sensor NLRs or the product of the R genes to cause the immune response. It's a very complex network that has emerged in the asteroid plants and has really exploded in some of the Solanaceae, for example, where it's up to half of all the NLRome is part of this network. One of the most exciting observations about the network is that it is phylogenetically structured. So you can see here that we have the NRCs being one clade and all the sensors being a sister clade. So it does look like they evolved from an NLR pair, and we predict that this happened about 100 million years ago or so, that when this expansion, massive expansion really, because you can see the sensors, they evolved to detect anywhere from bacteria, viruses, or mycetes, aphids, nematodes, and so on. So it's really over 100 million years, they acquired a lot of new specificities. So one of the observations we made when the ZAR1 structure was published is that the N-terminus of ZAR1, that alpha helix, as I told you, that flips out and forms a funnel-like structure, well, that's conserved across NRC. So you can see here NRC4 aligned with ZAR1, and that N-terminus is really similar, even though one protein is from Solanaceae and the other one is from Arabidopsis. And it turned out that this sequence matches a motif we call the MADA motif, which is present in about 20% of all the CCNLRs. Shown here in red in the tree on the right side. So these are all the NLRs that have the MADA motif, and we show that it's functionally conserved across diverse NLRs, from dicots to monocots. PIC2, for example, is a rice NLR. So this was really interesting because it told us that this motif has been retained throughout evolution, throughout long evolutionary time in many CCNLRs, and is obviously functionally important. But perhaps the most interesting observation was that if we look and zoom in onto the NRC supernetwork, or superclade, if I may say, then you can see that the great majority of the NRCs here are in red. That means they have the MADA motif. And again, we showed that several of these are functional. but none of the sensors, including all those famous R genes, have a MADA motif. So the MADA motif has degenerated over time in these sensor NLR. So they specialized into sensing, so they don't have the capacity to execute the cell death anymore. They just have degenerated MADA, and they're incapable of triggering the cell death on their own. So this is really how we ended up with this model that throughout evolution, the sensors and the helpers have specialized and then diversified into this network. And the sensors have degenerated MADA motif and also have these N-terminal integrations, which also will preclude functioning through resistosome type model. So now, new project, another lockdown project. Well, what about singletons? What about ancient and high fidelity, if you like, or conserved NLR? ZAR1 is one of these. So we started sort of looking at ZAR1, where it's conserved and all that, designed the pipeline to really dig out as many ZAR1 as we could. And we found that ZAR1, shown here in red, forms a very tight clade of core orthologs, essentially, if you like. So in this species, in quite large number of species, we have ZAR1, 1 or 2 copies of ZAR1, which are basically behaving like orthologs. There are a couple of subclades that emerge from ZAR1. ZAR1 sub, for example, is an early duplication, presumably single duplication of ZAR1 that in the eudicots resulted in a paralog family. But we have 88 angiosperm species that carry a ZAR1. This is a tree of the ZAR1. This is actually a tree based on the full length NLR or full length ZAR1, which usually is not possible, but ZAR1 is conserved enough that we can do that. And you can see that we can find it even back in some monocots in taro actually, and magnolit and stout camphor. So it is quite ancient, and we trace it back to over 150 million years. 150 million years ago, ZAR1, basically the phylogeny of ZAR1 traces the species phylogeny. So we concluded that ZAR1 emerged before monocots, magnolids, and eudicots split, which would be over 150 million years ago. So we call it a Jurassic NLR because basically when dinosaurs were roaming the earth, there was already some flowering plants that started showing up, and those flowering plants had ZAR1. So some of those giant Brontosaurus were actually eating probably some ZAR1 protein, and the resistosome was probably around at that time. So this approach allows us to develop a pipeline where we can now trace back conserved and divergent features of these proteins across really over 150 million years of evolution. So when we do that with the ZAR1 ortholog, that long series of 120 ZAR1 orthologs, what we find is a lot of red here in this barcode. This is out of a program called conserve. So red means conserved, and that means all these sequences, all these features have been conserved over 150 million years. And if we look at meme, what meme produces, we find the MADA motif. So all of these R1s are probably capable of executing cell death through a resistosome type model. They're probably oligomerized because the MBD-MBD interface is conserved. But maybe more interestingly is the kinase interface. I didn't mention that, but ZAR1 guards kinases of the RNA-seq family, so it directly binds its kinases, how it detects pathogens. The pathogen effectors are co-evolving with the kinases, but essentially these kinase interfaces are conserved also. So we conclude that ZAR1 has been guarding RNA-seqs way back since the Jurassic period, essentially, and has been probably functioning as a resistosome type protein for over 150 million years. This analysis also revealed an interesting new feature. So this was not known as being a conserved feature of ZAR1. But if you look at the underside of the resistosome, as shown here, you see this ring in red. And that ring corresponds also to this conserved motif that we pulled out with meme. And that ring is again really conserved, behaving as an essential feature of the ZAR1 resistosome. It's interesting because it's on the opposite side of the where the funnel structure inserts in the membrane. So what is the function of this? Is there a protein that docks in there? Does there additional features or activities of ZAR1 that we don't know of? This is work in progress. So we actually ordered the mutants, and we will be doing some experiments on this fairly soon, I expect. Okay, briefly, ZAR1 duplicated into the ZAR1 subclade shown in blue here. We think that's a single duplication event. And ZAR1 sub turned out to be a really interesting paralog family because it's quite degenerated. It's not as conserved as ZAR1. You can see here the ZAR1 sub conserved barcode there's a lot of blue there. So it's really diverse compared to ZAR1. And also these features like the MADA motif is gone, the MBD interface is different. The RNA-seq binding interface is also degenerated. So what we think has happened here is that ZAR1 sub has basically diverged functionally from ZAR1, neo-functionalized, I guess. Perhaps it's guarding a different type of proteins, maybe effectors directly, maybe another family of guardies, but it's certainly unlikely to be able to cause a cell death on its own because the MADA motif is gone. So probably ZAR1 sub became sensors. Are they functioning with ZAR1? Are they functioning with other helpers? We don't know yet, but this is again a great hypothesis coming from this EVO MPMI type of analysis. This is the summary. I don't really need to say much here because I'll be repeating myself, but essentially we have duplication here of the blue clade, the ZAR1 sub, some point in the eudicot evolution. Some families like the Caryophyllaris lost the ZAR1 sub, but ZAR1 sub have diversified also in many angiosperms, and they seem to have degenerated features of NLRs, typical of what we expect of sensor NLR evolution. So again, back to the take-home message, and to conclude, I think the message here really more than anything is that we should kind of go way beyond this uniform view of NLR structure and function. So first of all, NLRs can go in different evolutionary trajectories, and they can function in different ways. They can be specialized sensor receptors. Some of them have integrated domain. They can be helpers, or they can be multifunctional, essentially singletons, if you like, where the coevolution arm race is happening with the guardie or the decoy, not necessarily with the NLR itself. So I think it's really interesting to start sort of classifying and organizing the NLRomes based on this kind of understanding of the evolutionary trajectory and history. So I'll end there, and I want to thank many, many people. These are the folks that have done a lot of the work. So Shihan started the NRC network. Lida works on suppressors of the NRC network. I didn't talk about that, but Abbas works on structural biology aspects and biochemistry. Aki led a lot of the work on the MADA motif and more recently on ZAR1 evolution, along with Toshi and Mauricio in particular. Mao works on also suppressors of the NLR network, and we have collaborators, Toga Boskut, Mark Benfield, Ryohei Terauchi, and actually quite a few others. I should also mention here Yorgos, who did the RefPlant NLR and also drops a lot of ideas on pretty much everything, and Joe, who's been involved in a lot of the bioinformatics, and Adeline supporting all these teams. A lot of different people thank, but I'll stop there and I'll be happy to answer any questions. Thank you.
Great. Thanks, Sophien. So I'm going to start. So there are some questions that is not directly related to the talk, so I'll skip those ones and go to the questions that are relevant to the talk. From James, can the NLR from one species also provide resistance in another species? If so, how far?
Yeah, this is a really good question. And it was observed very early on that NLRs tend to be, or R genes, the sensors tend to be difficult to move across taxa. So they work usually in closely related species, but not far off. We believe that understanding the network architecture of the NLR of a given plant is really critical for moving them around, because if they require helpers, more often than not, you need to move the sensor with the helper to really get the resistance to be transferred. And we actually initiated collaborations with industry, the plant breeding industry. I think this is really relevant to them, because very often also you have an R gene that works really well in one particular background, but when you move it to an elite variety, for example, either stops working or doesn't work as well. And that could very well be explained by epigenetic effects caused by their basically network partners, for example, the helpers. So it's a very good question. And the answer is that it depends. So if it's a singleton, it probably will work if you move it around, but if not, then you need to know the structure, the network structure.
Thank you. Question from Pavitran. So some plant pathogenic nematodes are supposed to have loci with hypervariable domains, which generates different variants of the same effectors in individual nematodes. So this gives advantage in terms of infection. Are there comparable examples in the microbial world with the same effectors differing among strains, accessions, and how has it shaped receptor evolution?
Yeah, so I think a feature of effectors in any system is that they are rapidly diversifying and exhibit all types of variations. Sometimes it's just in terms of sequence variation, accumulation of amino acid diversification, but in other cases, it's also based on repeats and so on. I think if you want an example of hypervariable repeats or hypervariable domains, then you need to go no further than the tile effectors, which are actually based on repeats, and there the mechanism is very well known. So evolution, whatever works. So I think the simple answer, Pavitran, is that yes, there's pretty much a lot of solutions to this problem of diversification of effectors.
Thank you. Question from Ralph Hukeloven. Can we distinguish helpers and singletons or standalone NLRs by sequence only?
That's literally the million-dollar question, because this is exactly why we build collaborations with industry. And I think the answer to that is yes, to some degree. So for example, I guess the presence of a MADA motif or the absence of a MADA motif is indicative of a CCNLR that is probably unlikely to signal on its own. Like I just told you, the hypothesis we basically came up with for this R1 sub, right? So I think to some degree, yes. I think there's multiple factors that can be integrated into these analysis. It becomes really systems biology type of approach. And I think we have data sets like evolutionary rate, presence of certain sequence motifs, expression data, co-expression data, phylogenetic distance, and so on. These are all things we are actually doing at the moment. And actually the ZAR1 exercise is a good one, because that's a simplified version where we can make simple predictions about the classification essentially of these NLRs.
From Shumei Chen, when did tier type NLRs arise?
Oh, good question. Very complicated one, to be honest. First, because I don't directly work on tier NLRs, so I'm not necessarily following anything, but I don't know if you've been seeing some of the work coming out of the bacterial immunity field, and there are tier proteins involved in bacterial immunity too. So it's really interesting to kind of understand where actually how the tier domain in itself, independently of whether it's linked to an NLR or not, like when did it start becoming involved in immunity? Is that single ancient activity and then they got recruited into NLRs, or is it more recent? But as you probably know, there are no tier NLRs in monocots. So presumably the simplest explanation that they arose later, but I'm not an expert of tier NLRs. We haven't directly looked ourselves. So I think it does require deeper phylogenetic analysis, maybe also with early diverging plants to answer that question.
Cool. And a question from Oliver Arts. What do you think could have triggered the explosion of new sensors helpers about 100 million years ago?
Very good question. I really don't know. It's very hard to know, but I skipped those slides, but they're in the paper. They're in Shihan Wu's paper from 2017. If you look at the Caryophyllalis, like sugar beet, for example, then it's actually a very simple situation. There's actually only 3 NRC network genes, and they're in one cluster. One is a helper, 2 are sensors. And if you go to other Caryophyllalis, you can find actually single NRC type genes. So clearly they started as single gene pair and then they exploded. I really don't know, but I suspect the fact that it's, I also didn't mention that, but the fact that it's a genetically dispersed network. So if you look at tomato, for example, the genome is pretty good. The NRC network, they're all over the place. There are connections between genetically unlinked genes on different chromosomes. So I think there must be a feature in the way these NLRs in the NRC network function that allows them to actually work together even if they're genetically unlinked. And obviously, if you add the GARDs and decoys, you have another layer in the network where you have even more genetic dispersal, if you like. So I don't know the answer, but I suspect there must be a mechanistic feature that allows these NLRs to actually spread throughout the genome, that maybe others, like the pairs that are stuck together for a long time, don't experience.
And a question from Amandeep Kaur. So how about the diversity of ZAR1 between monocots and dicots?
Yeah, so that's a very good question. That was part of the analysis I showed you. So the 120 orthologs, that includes monocots. I have to also say that we only found it in a single monocot species. So most of the monocots have lost ZAR1. You don't find it in rice, wheat, and so on. But you find it in taro, so Colocasia. Actually, by the way, I just had Colocasia for lunch yesterday in an Indian restaurant here in London. So it's really taro is such a delicious kind of potato of the tropic. But Colocasia has a ZAR1. So, but that was part of the analysis. So it has all the features of the key features of Arabidopsis ZAR1. You do find them in Colocasia ZAR1, which is from the phylogeny probably the most distant to the eudicot ZAR1s. So it does look like ZAR1 was already, before monocots and dicots diverged, there was ZAR1 that was functioning just like the Arabidopsis ZAR1. That's the conclusion we draw from this work.
Cool, thanks, Sophia. Maybe I can ask one last question. So you showed us that NLRs are quite modular proteins, so they have clearly defined domains. So when you look at your phylogeny, do you see certain regions that may be responsible for subcellular localization?
That's a good question, and I think the subcellular localization, I think this approach of looking for conserved features could be a way to actually pull out motifs that maybe are important for subcellular localization. Expect them to be, I guess, on the surface. So maybe that underside pattern we found that's conserved, maybe that's involved in trafficking and maybe removing it from the membrane after it does the job. To answer you, I think the simple answer is we find a lot of motifs that are conserved that we don't know anything about. So there's a lot of things to discover there. But maybe my more general message to this audience is that whatever protein you work on, if you have actually a core ortholog series, the approach we produced here is actually a pipeline that's very straightforward. Meme, conserve, and then mapping on the structure. So if you have a core ortholog series and you have structural information, you can really learn a lot about your protein by doing that. The key, of course, is that you have an ancient protein, which, of course, in the case of NLRs, most cases you don't even find it in the next cultivar or next accession. So you need to have conservation over a long time, and then you can actually do this sort of analysis. So I encourage many of you to do that. We'll be posting the preprints on ZAR1 evolution pretty soon, so you can check the methods, or you can email me or contact me.
Great. Thanks, Sophien. I don't see more questions, so we can move on. Thanks again. So our next speaker is Philippe Carella, and he's a group leader at John Innes Center, and he will be telling us about continuing the evolution theme and telling us about earlier immune system and immunity in Markantia.
So, right. So plants colonized land at least 460 million years ago, and they would have occupied a pretty harsh environment. They evolved from freshwater charophytic algal predecessors, which moved from water onto a rocky soilless substrate that would have been very scarce in mineral nutrients, so very challenging. And at this time, this environment was teeming with terrestrial microbes that evolved 500 million years even before this. So it was a pretty hectic environment, let's say, and definitely this is well before there were dinosaurs or people. So unfortunately, this is the only dinosaur cartoon that I can show today. Since then, plants have evolved into a number of lineages that we're familiar with now, from early divergent bryophyte lineages, which are non-vascular, non-seed plants, to the early vascular lycopods and ferns, to seed plants and ultimately flowering plants. The diversity of plants we know interact with a very diverse range of microbes. we're most familiar with studying model angiosperms like Arabidopsis, but there's much less known, let's say, about bryophytes, and in particular Marchantia polymorpha, which is my main model organism. And the theme of my research, the overall question, is to use the diversity of plants to understand how plant defenses have evolved over the 450 million years. So, like I said, to do this, I'm using Marchantia polymorpha. It's a very accessible and informative early divergent land plant lineage. This is a picture of some Marchantia growing outside. So it grows laterally along the surface and along the substrate rather. On the surface of the plant, it forms these gemma cups. So these are like these little bowls that inside there are little disc-like propagules called gemmae. So when water splashes down, when it rains, these are displaced onto the soil, and then these will sort of germinate and grow into a clone of the mother plant, let's say. This is about 1 week's worth of growth, where you can start to see the tissue pattern developing here. So if you take a cross section through here, you can see the sort of breakdown of how the tissue is organized in Marchantia. So at the top, we have a photosynthetic layer, which houses these individual air chambers. If we look closer, we can see that these have a single open pore, and inside there's an intercellular space with these photosynthetic filaments, very carbohydrate rich. Underneath this, we have storage cells, non-photosynthetic parenchymatous storage cells and oil body cells. Underneath, again, we have rhizoids, which are root hair-like structures. So Marchantia is dioecious, so we have male and female individuals. Here you can see the sexual structures that were induced by far red light. So females on the left, males on the right. We're very lucky that there's a great Marchantia community, and we have a good, very good sequenced genome, lots of reliable transformation protocols, whether you're working with thallus or sporelings, gemmae, and we can do genome editing and transformations rather quickly and efficiently. So it's really a good model to work with, and I'm happy I'm working with it. To probe plant defenses in Marchantia, we're using Phytophthora palmivora. It's a broad host range oomycete pathogen. It can be transformed, which is great. We can express fluorescent proteins that allow us to track it at the cellular level. So here there's intracellular infection structures, Haustoria. We can also track it at the tissue level, and palmivora can infect a number of model host plants and can colonize both leaves and roots of these plants, which is great. So it seemed like a good candidate to start with if we were going for a pathogen that's going to infect an early divergent land plant. So one of the first things I did was, just mix the two of them together. So I dropped spores of palmivora on top of liverwort thalli. And you can see from a time course of 1 to 7 days that the plants are succumbing to infection. These droplets, the pathogen starts growing and then spreading along the thallus until it takes over the entire plant. All of the isolates that I've tested, different palmivora isolated from different host plants around the world, all seem to be able to infect Marchantia very well. And they all do so by colonizing this upper photosynthetic layer. So here the pathogen is labeled with red fluorescent protein, and it's mostly limited to the photosynthetic layer here. And this is at around 3 days after infection. I'll say that towards the end of the infection, 7 days, you start to get necrotrophic hyphae that push through, and it'll eventually eat the whole plant. But the early biotrophic phase seems to occur predominantly in this upper photosynthetic layer. So characterizing the interaction, we started with some RNA-seq analysis to see what was going on, both on the pathogen side, but I'm going to talk about the plant side. So at 1 day after infection, you can see that there were spores here on the surface of the plant. This is an air chamber, and the hyphae have germinated along the surface. We also looked at 2 to 3 days after infection, where the pathogen is starting to colonize individual air chambers. And then later at 4 days after infection, where mostly the plant is colonized, and then sporangia are developing, so the pathogen is completing its life cycle. And for the RNA-seq analysis, we compared infected versus mock-treated plants at each of these time points. And we can see that there's an increase in certain infection-associated genes starting from around 2 to 4 days after infection, and some that are showing up more later towards 4. And we also did some proteomics in collaboration with Hiro Nakagami, his lab in Cologne. I'll say Hiro did it, and we found some really reliable markers that are upregulated in both the RNA-seq and the proteome data sets. There's about 187 loci. And from these, we see some typical players. So we see that liverworts can deploy pathogenesis-related genes that are very typical of angiosperm pathogen interactions. So for the MPMI crowd, you're very familiar with these PR genes, small secreted proteins, and these families here. So Markantia does have PR1 protein, PR2, but the PR gene that was really highly induced during infection was PR6, a protease inhibitor. And here's a reporter line where we've hooked up the PR6 promoter to TD tomato localized into the nucleus, and we can see in mock-treated plants, the expression is pretty much off, and in infected plants, this is a YFP-expressing Phytophthora. Even though this one air chamber here is colonized with the pathogen, we can see strong induction of this marker gene in the colonized area and the cells adjacent to it. But if we're looking at the tissue level expression profiles using GUS reporters, we can see that individual air chambers here and only those areas that are colonized are expressing PR6A. So it's a bit of a more localized response. And with some other PR genes, we can see a more systemic induction of gene expression across the entire body of the thallus. So irrespective of where the pathogen is. So it seems that liverworts do have mechanisms for localized versus systemic responses to infection. And we looked at a number of other gene families typical of plant microbe interactions, and what we could see was these microbe-responsive liverwort genes in Markantia typically have a counterpart in flowering plants. So these genes are likely conserved across all land plants. But the real question was whether during infection with Phytophthora palmivera, whether liverworts and angiosperms would share a common response. So are they inducing the same sets of genes? So to do this, we did some more RNA-seq. We did it in Nicotiana benthamiana. And here we've compared the two RNA-seq data sets. So since there's a large divergence time between these two plants, we're looking at single copy orthologs only here. It's a bit complicated, but I can walk through it. So on the y-axis, we have log twofold change of the Markantia ortholog. X-axis is the fold change of the Nicotiana ortholog. So anything that's present in this top right quadrant is induced during infection in both hosts, or in both plants. Bottom left is suppressed in both, and then these are opposing genes. So we saw that most genes that were induced, or most genes that are differentially expressed, were occurring in the same direction. So there's a similar transcriptional response to infection. Looking at these types of genes, the one that stood out really were phenopropanoid, associated with phenopropanoid metabolism, let's say, where there was a number of shared orthologs that were induced in both Nicotiana and Markantia. And something that was interesting, and first I'll just say that this was one of the more striking results that we saw. So here, this is showing differential expression of all transcription factors that are associated with response to Phytophthora infection. So there are 30 genes here, 30 transcription factors, and they are belonging to about 8 different families. So this is it. It's just 30 transcription factors differentially expressed. And in terms of phenopropanoids, we saw a single MIB transcription factor that was really strongly expressed. And this is relevant because we know that R2, R3 MIBs, like PAP1 and Arabidopsis, are associated with flavonoid, anthocyanin, and phenopropanoid biosynthesis. So it gave us an idea that maybe this is happening in Markantia. So when we overexpressed this MIB transcription factor, MIB14, our thoughts were confirmed here. We saw these plants are stunted, but they're accumulating really high levels of a dark red purplish pigment. They're severely stunted, actually. These are 2 months old compared to a 2-week-old liverwort here. And around this time, at first you think you're scooped, and then you realize that this is actually one of the best things that could happen. But 2 independent groups published on the function of this MIB transcription factor, and they showed that when you knock out MIB14, you lose this purplish pigment relative to wild-type plants, and this is during nutrient stress, nutrient deprivation. And they could also overexpress this transcription factor and see that you get colorless flavonoids accumulating in the cytosol and also this dark red purple pigment in the cell wall, which they eventually showed was rickeonidin A, a very unique type of anthocyanidin-like chemical. So we took advantage of their transcriptome data and we compared their expression analysis of MIB14 overexpressors compared to wild type, and we compared this against those genes that are induced by Phytophthora palmivera in wild type plants. And it was quite striking to see 191 genes that were shared in common, and this represented around 25% of all infection-induced transcripts in wild type plants. So these could be directly tied to MIB14. Of these, a lot were associated with phenylpropanoid biosynthesis, so chalcone synthases, phenylalanine ammonia lyases. So again, this confirmed to us that phenylpropanoids were likely involved in the response somehow. So instead of looking at genes, we took a better look at the plants, and we could see during infection that we got this purplish pigment accumulating around the walls of air chambers during infection that we don't see under mock conditions. And when we looked a little bit closer, we could see at these deposits, so shown here in the arrows, these purplish deposits, generally there was less fluorescent pathogen in these areas. But to be more sure about this, we took a closer look. So we used MIB14 mutants that were generated by Hiroyoshi Kubo's lab. We compared infection profiles of the MIB mutants to the wild type plants. We characterized how the pathogen was growing by qRT-PCR, and we saw in 2 independent knockouts of MIB14 that pathogen biomass was really accumulating here. So these plants are much more susceptible to infection. And this susceptibility was associated with an inability to induce phenylpropanoid biosynthesis genes like phenylalanine ammonia lyase, chalcone synthase, and chalcone flavonoisomerase-like transcripts, which are all really well induced in wild type plants. So we wanted to go the other way and to induce pigment accumulation in Mercantia and see if this had an impact on resistance. We clearly couldn't use these plants because they're so stunted, but mostly because they eventually just died. They were really quite unhappy. So we wanted to do conditional gene expression. And in the literature, there is a way to do this with heat shock promoters. So here we're showing the Mercantia heat shock promoter expressing GUS. And after heat shock, you can see a really strong GUS response. And in our conditions, in our hands, this just didn't work the way that we expected, but it worked in quite a wonderful way, let's say, where we saw just spurious activation of the heat shock promoter, but it resulted in these sectors of pigmented versus unpigmented cells. So this allowed us to compare pigment accumulation on the same plant, which was quite lucky, I'll say. So when we tested this, I did this by dropping some spores on the pigmented regions of the liverworts and just seeing what the pathogen was doing. And you could see after around 5 days, pathogen is pretty much colonizing only this unpigmented green part of the plant and is avoiding the purple areas. And you can see this by the fluorescence of the 2D tomato marker, where around the edge where the pigment is, it's really avoiding this. And we also quantified this with qRT-PCR, where we see pathogen biomass in the green parts here in wild type plants and really quite reduced in the purple areas of the heat shock MIB14 lines. So taken together, this is showing that liverworts really share common responses with angiosperms. We see spatially distinct activation of PR proteins. We're also seeing common shifts in host transcriptional profiles, and we see strong metabolic reprogramming that's centered on these phenylpropanoids. And this is a great example of how we can use evolution to understand fundamental biological processes. However, I will say that we've just focused on 2 ends of the spectrum here. We've focused on Benthiae and Mercantia. So we can't really say whether we're seeing similarity that's due to conservation or just convergence. So moving forward with my group here at JIC, I want to take a look at some other, let's say, underrepresented plant lineages to sort of tackle this question and to better define the evolutionary framework that's underpinning plant defense in land plants. And here we can also explore some well-known plant defense, plant immunity pathways. And Mercantia is particularly useful for this because it has a number of cell surface receptors that are involved in pattern-triggered immunity. We can also see pathogen effector activity in liverwort cells, which is suppressing plant immunity. And related to Sophien's talk, Mercantia does have NLRs. It has a reduced set of these NLRs. And we don't know yet what they're doing, but we can start to ask questions of whether they're contributing to effector-triggered immunity. And whether non-vascular plants have any inclination towards systemic resistance, we do see sort of localized versus systemic-wide responses in Mercantia, but this is an open question, and it's quite interesting. And I'd say that Mercantia has orthologs probably capable of synthesizing at least some, if not all, of these compounds here involved in systemic acquired resistance. So these are the types of things that I'm going to be looking at in our new evolutionary phytopathology group here at the John Innes Center. I started August 2020, about a month ago, and this is what JIC looks like, and right, that's backwards. This is what JIC looks like, and here is where the liverworts will go. And with that, I'll just acknowledge everyone in Sebastian Shornack's lab. Seb is a great advisor and really supported me throughout this, and part of the reason I got the job is because of him, I'm sure. Everyone in green was directly involved with the project, and here Hiro Nakagami and Hiroyoshi Kubo have contributed to the results as well. And thanks for your attention. Happy to take any questions.
Great, thanks, Phil. So there is already a question, so I can start with that one from Pavitra Narayanan.
Sure.
How much did your RNA-seq study reveal about the role of phytohormones, JA, SA, etc., in defense against, he says P. infestans, but I guess P. palmivora?
Yeah, so I would say it is kind of difficult to tell. We don't really know everything about SA signaling yet, but MYC2, which is associated with JA, and Mercantia doesn't make jasmonic acid, it makes dinor OPDA, which is a sort of modified precursor of JA, but it does make a JA type molecule, and I think there are signatures of its induction and it having a role during infection. So I would, yeah, it's happening.
Okay, so there is one question in the chat. So from Nico Cecchini, did you find NS-LTP in Mercantia? I assume you would know this.
Yeah, I'm aware of Nico. Hi Nico. So yes, there are, so these lipid transfer proteins, so it's non-specific lipid transfer proteins, and they can be induced during infection. Some of them play signaling roles in systemic acquired resistance or a role in basal immunity as well. There is an NS-LTP that's induced strongly during infection. These proteins are difficult to work with. I tried to do a couple of things, but yeah, there's still more to be done. But it's induced.
Okay, question from Ralph Hückelhoven. Does Risionidine A cause the resistance or just mark the hostile environment on the plant?
So it's difficult. The short answer is we don't know yet. I think it's probably both things. So MID14 is inducing Risionidine A deposition in the cell wall, which is thought to play a role in either cell wall strengthening or ROS scavenging, some redox activity as well. And at the same time, you're also getting an overaccumulation of these colorless flavonoids and other phenopropanoids in the cytosol. So at the moment, we can't really determine whether it's the riccionidin, the purple pigment, or it's these colorless flavonoids that are having the main effect. So until really we have biosynthetic mutants towards the end of the pathway where we don't get riccionidin A, we can't know for sure yet. But let's say that the pathogen has a much harder time penetrating into host cells. So I'd be surprised if riccionidin A at the cell wall isn't having an effect.
Okay, from Gustavo McIntosh, do you see cell death associated with the defense response against oomycetes?
So, so it's hard to tell at the moment. I've tested, so it's part of the reason that I wanted to test a number of Phytophthora palmivera isolates to see if we can sort of recapitulate these compatible, incompatible interactions and see if we can trigger ETI using a pathogen. But so far, every strain of Phytophthora palmivera that we've tested can colonize and cause disease on Marcantia thalli. Phytophthora infestans can't, and it appears that it might be associated with cell death, but we need to look at this in a little bit more detail.
Great. From Silvia Ramundo, any idea about why photosynthetic cells get preferentially infected? Do chloroplasts play any role during this infection?
I would think so. So we did try to infect the underside, the rhizoids, and we really didn't see anything there. Really, we only saw a good infection from the top down. And a sort of simple answer is that these photosynthetic filaments are really rich in carbohydrates, so they might just be, you know, juicier and preferred by the pathogen. So, yeah.
Okay, and from Yunkin Yu, considering MIB transcription family, transcription factor is a very large family in angiosperms, I wonder how many members are there in liverworts?
Yeah, so the R2, R3 MIB transcription factor family, I think, I don't remember the exact number, but it's on the order of tens, not hundreds, like you see in angiosperm. So it might be like 30 or so. But what was striking was really we just saw two that were differentially expressed during infection in Marcantia. One that was strongly upregulated, which was MIB14, and the other was downregulated, which is MIB2. And MIB2 is also playing a role in biochemical, well, I think flavonoid and other compound biosynthesis. So it's really quite streamlined in liverworts compared to angiosperms.
And from Xunyuan Xiao, do you know if liverworts have any obligate biotrophic pathogens?
Obligate biotrophs. So far, there's not much described in terms of obligate biotrophs. Mostly just sort of the approach that we were going with of taking a well-known pathogen with a broad host range and using it to probe immunity in Marcantia. Others have looked for pathogens that are naturally associating with liverworts out in the wild, more of an ecological survey. And in those cases, you tend to find more necrotrophic fungi and saprotrophs. So we don't have anything just yet, to my knowledge, anyways.
Cool. From Dimitri Lepin, cool microscopy. Could you speculate about the steps plants took to build up Marcantia-like immune system from the blocks present in algae?
Yeah, so there is quite a bit of work showing a complex accumulation and biosynthetic pathways for phenopropanoids and flavonoids in algae. So I think certainly that was part of it. Let's say COI1 and JA or OPDA-like responses were derived from algae as well, to my knowledge. And yeah. So I think there are quite a few components and definitely the biochemical side of things. There is an origin there from algae onward. And some of the pathways or the mechanisms contributing to NLR-mediated immunity have orthologs at the very least in algae. So I think, yeah, there's something there.
Cool. And maybe if I can ask a question on this
Sure.
conservation convergence thing. So can you enhance immunity, let's say, against infestans or palmivora in Bentamiana by overexpressing either the Marcantia transcription factor or the Benti homolog? Have you tried just expressing it and infecting with pathogens to see if it also enhances resistance?
So yeah, so I've tried to express the MIB14 and Benti, didn't have much luck, and to be honest, I didn't expect. Well, the pigment isn't made in angiosperms, anyways. It's sort of unique to liverworts, and I think there's a couple other plants. But certainly, if you overexpressed a MIB that is leading to phenylpropanoid accumulation, you'll see enhanced resistance to at least a subset of pathogens. So these things are known to play roles in providing resistance. But that's part of the goal, to use the diversity of defense strategies that are employed by land plants to ultimately transfer and protect crops. That would be something useful.
One last question from Pavitran. Does genomic simplicity of Mercantia allow easy infection by pathogens in terms of time taken, etc., compared to higher plants? Yeah, so it does. Maybe let's say the simplicity of the system is a bit, not oversold, but maybe the message is taken too strongly. Certainly, when we're talking about transcription factors, there's a drastic reduction or simplicity there. There's far fewer numbers. But when we were looking at genes involved in phenylpropanoid biosynthesis, for instance, it's massively expanded in liverworts. So it just depends what family you're looking at. So there's a potential for things to be complicated in a simple system. Great. Okay. Thanks, Phil. And thanks everyone for really good questions and lively discussion. Before I close, I'll leave it to Katie to see if she has anything to say.
Yeah, I would just like to say thank you to our speakers and to you, Yasin. You did a great job with the moderation. It's pretty cool to feel like part of the extended Kamoun family. So our next webinar will be, I put a link to it in the chat. It'll be in the Australian time zone. So if you're able to be awake then, feel free to join live, but you can also check it as a recording afterwards. Thank you all for joining and for participating.